In this video, we will discuss membrane structure and function in more detail. The plasma membrane, as we already know, is the boundary that separates a cell from its surroundings. The plasma membrane exhibits what we call selective permeability. What this means is it allows some substances to cross more easily than others, and it actually regulates the passage of materials into and out of the cell, which, as we'll find out, is a very, very important characteristic. Cell membranes are made up of two primary components: phospholipids, which we've talked about a little bit before, and proteins. Now, phospholipids are what are referred to as amphipathic molecules. That's just a fancy word for saying that they have two different characteristics. And phospholipids have both hydrophobic and hydrophilic regions, which we've talked about before. Because of this amphipathic nature, what phospholipids can do is they can form a bilayer and can exist as a boundary between two aqueous areas, as we know cells exist in. They have a fluid inside and they live in a fluid outside. We refer to the cell membrane as a fluid mosaic. That is because the proteins and the lipids in it can move around. That's the fluid part. And we refer to it as a mosaic because it has a number of different components: the phospholipids, proteins, cholesterol, a variety of other things. We can see this here in a model. Notice that there are multiple components in the membrane. Here's the phospholipid bilayer. We see with the hydrophobic tails inside and the hydrophilic heads outside. Here we have a protein that spans the entire length or width of the membrane. Here's one that we find only on the inside. And then we notice the extracellular matrix that is bound to some of the proteins in the membrane. As we mentioned before, the phospholipid bilayer, or the cell membrane, is a fluid structure. And as I mentioned, what that means is the lipids and the proteins can drift laterally. That means they can move around in the membrane. Now, they don't move around very far, but they are capable of moving around somewhat in the membrane. Again, that's the fluid nature of the plasma membrane. The protein structures make up the mosaic. So again, we call the membrane a fluid mosaic model. And there are a number of different proteins found in the cell membrane, and they have a number of different functions. First of all, there can be peripheral proteins, which are proteins that are found only on one side of the membrane, so they're bound to the surface, so they don't go all the way through. Integral proteins, on the other hand, go all the way through the plasma membrane, and they are also called transmembrane proteins because they go all the way through. Here's a integral protein. You can see here's the outside of it, the extracellular side, with part of the protein sticking out. Here's the part that is found within the membrane, and then here's the cytoplasmic side sticking out into the cell. There are 6 major functions of membrane proteins: transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to the cytoskeleton and extracellular matrix. Here's a diagram of those functions. You'll need to check those out on your own. We're not going to spend a lot of time on those in this video. As we've already talked about, structure typically dictates function when it comes to biological molecules, and the cellular membrane is no exception. Based on the way that the cellular membrane is put together, with the hydrophobic tails of the phospholipids pointing inside and the hydrophilic heads pointing outside, it's that structure that leads to the selective permeability of the membrane. The proteins, also, if embedded in the membrane, also contribute to the selective permeability. And remember what that means. It means that the membrane can regulate the passage of things into and out of the cell. Hydrophobic molecules can dissolve in the lipid bilayer and pass through the membrane fairly rapidly. They can actually get through the hydrophilic parts and then travel through the hydrophobic parts. By contrast, hydrophilic molecules do not cross the membrane easily, and they need some help. And we'll talk about that more as we get further through this lecture. Transport proteins are what allow the passage of hydrophilic substances across the membrane. These are definitely integral proteins because they span the entire membrane. There are 2 kinds. There are channel proteins and there are carrier proteins. Channel proteins have a hydrophilic channel that serves basically as just a doorway or a tunnel for things to flow through. One of the really important channel proteins in the cell is what are called the aquaporins, and they allow water, which we already know is a very hydrophilic or polar molecule, to pass through the membrane without any problems. The other transport proteins are called carrier proteins, and they are a little bit more specific than the channel proteins. Typically what happens with carrier proteins is they will bind to a molecule, change shape, and then send the molecule through the membrane. The process of movement through a membrane is called transport, and we're going to talk about two different kinds of transport in general. The first is called passive transport, and this is the movement of stuff across the membrane without any energy input on the part of the cell. Okay? The first type of passive transport is called diffusion, and diffusion is the tendency for molecules to spread out evenly into an available space. Think about when you're cooking in your house. The smell is more concentrated in the kitchen, but after a while, the smell of the food gets throughout the entire house or apartment or wherever you live. That's diffusion. Okay? Diffusion usually results in equilibrium, where the molecules are distributed equally and evenly in the available space. Here's an example of diffusion. Let's say we've got an aqueous solution with a membrane separating it. We add some sort of molecule to one side. Let's say we add a dye here. So as you can see on the left side, there's more molecules. Okay? In order to reach equilibrium, the molecules are going to want to move from the left to the right or through the membrane. After a while, we will get to equilibrium where the molecules are moving equally across the membrane and they're evenly distributed. Now that's the diffusion of one solute. The same thing can happen with two solutes that may be unevenly distributed. Ultimately, they're both going to flow equally through the membrane and achieve equilibrium. Now, what causes substances to move from one area to the next? Basically, substances will move from an area of high concentration to an area of low concentration. This difference in concentration from one area to the other is called a concentration gradient. So in diffusion, again, molecules move from a high concentration of the molecule to a low concentration in order to achieve equilibrium. This is what we would call moving down the concentration gradient. And again, this is a type of passive transport because no energy is necessary to make this movement happen. Diffusion is a specific type of passive transport, as is osmosis. Osmosis is a passive transport of water across a selectively permeable membrane. We identify this as a specific type of passive transport since water balance in a cell is critical to its function and survival. So what happens is the water is going to diffuse across a membrane from a region of low solute concentration to a region of high solute concentration. So to put this another way, water will follow solute. So water follows a similar pattern of movement as diffusion does. So wherever there's more water, it's going to move to an area where there's less water in order to equalize the amount of water on either side of the membrane. In order to understand osmosis and the way water moves in and out of a cell, we must understand the concept of tonicity. Tonicity refers to basically the amount of solute in a given area. We could also say it's the ability of a solution to cause a cell to gain or lose water. In an isotonic solution, what that means is the solute concentration is the same on both sides of the membrane. So in a cell, it's the same inside the cell as it is outside the cell. So there will be no net movement of water, meaning more water won't be going into or out of the cell. In a solution that is hypertonic, the solute concentration is higher than it is in the other area. So for example, if we placed a cell into a hypertonic solution, the outside solution would have more solutes dissolved in it than the cell would. Okay, hyper means higher, so that's where we get this definition. What that means is the water is going to leave the cell in order to try to equalize the solute concentration. By contrast, a cell that is placed in a hypotonic solution, or one that contains less solute than the cell, will cause the cell to gain water, and the water will flow into the cell. Here's some examples of that. In an animal cell in a hypotonic solution, there's going to be more water outside the cell than inside, so consequently the water will flow in. What that can cause to happen is it can make the cell burst because the cell takes in more water than the membrane can handle. In an isotonic situation, the same amount of water is flowing in as is flowing out, and this is the normal or equilibrium situation. In a hypertonic solution, remember that there are more solutes outside the cell. What that means is there's less water outside the cell than inside the cell. So the water is going to want to flow outside and it's going to cause the cell to shrivel. A similar kind of thing happens in plant cells. The difference with plant cells is their shape is maintained by the cell wall, so they aren't necessarily going to burst. If you don't water your plants, the plants enter into what is called a plasmolyzed situation, where the membrane literally collapses from the cell wall and they become limp and they wilt and they can eventually die. Again, if a cell is in a hypotonic solution, it's going to swell. So in a plant, we would call this a turgid or firm situation. This is the preferred situation for a plant. It doesn't want to be in an isotonic solution because it can actually become limp. It wants pressure inside, so it wants more water inside in order to be healthy. As we said before, in a hypertonic environment, the plant is going to lose water and the membrane literally pulls away from the cell and can cause the plant to die eventually if you're not careful. And this is called plasmolysis. Another type of diffusion is called facilitated diffusion. This is one in which transport proteins move molecules that couldn't ordinarily pass through the plasma membrane. Now, this again is a passive process, so it doesn't require any energy. And transport protein includes channel proteins and carrier proteins. A good example of this would be the carrier protein that moves glucose into the cell. Remember that glucose is one of the primary energy sources for cells, so it wouldn't make sense and it would be inefficient for a cell to have to use energy to get glucose inside of the cell. So it travels through a transport protein. We've talked about channel proteins already. They provide basically hallways or corridors or tunnels for molecules to travel through, and an aquaporin is a specific type of one. Other types of channels are ion channels that allow charged ions to transfer in and out of the cell without any energy input. And then we mentioned carrier proteins that undergo a change of shape that allows them to move things into and out of the cell without any energy need. Here's an example of a channel protein. Stuff can flow in without any energy. And here's a carrier protein that the molecule that it moves is going to bind to the carrier. The carrier is then going to change shape and it's going to send the solute or the molecule into the cell. Now, in some cases, the cell needs to move molecules against the gradient, or it needs to move molecules that may require a little bit of energy in order to do that. We call this active transport because it utilizes energy. Okay, so any type of transport that utilizes energy, or a situation in which the protein that moves the solute through the membrane and requires energy is active transport. Typically, it's also moving substances against their gradient or up the gradient. Think of it as pushing your bike up a hill. It requires energy to do that. And just like other transport types, active transport is performed by specific proteins in the membrane. One of the primary kinds, or one of the main examples, is what's called the sodium-potassium pump. What this does is it moves sodium into and out of the cell actively in order to maintain their gradients. Okay, here's the example of the sodium-potassium pump. The sodium will bind to the pump, energy is used in the form of ATP, and then the protein pumps the sodium outside. Then, by contrast, potassium will bind to the protein, and then it will open up into the cell and move the potassium into the cell. So in cells, or in your body, the concentration of sodium outside the cell is higher than it is inside. By contrast, the concentration of potassium inside the cell is higher. This is what's called a membrane potential, and it's really, really important for how your nerve cells function, how your muscles function, stuff like that. So this is a really important active transport mechanism in cells. Again, what this produces is the membrane potential, which is the voltage difference across the membrane. So there's actually an electrical charge difference across the membrane. And again, this is really important for muscle contraction as well as nerve signaling. The last type of transport is bulk transport, and this is where the cell needs to move molecules that may be just too big to get through the lipid bilayer or too big to get through transport proteins. And this also requires energy, so it's considered a type of active transport. The first kind is called exocytosis, and this is where a cell releases things into the environment. The prefix exo means outside of. This is a secretory function for lots of cells. So when cells need to secrete proteins or enzymes or whatever, they form vesicles that usually come from the ER and then the Golgi, and then those vesicles are going to fuse with the cellular membrane and move the molecules outside the cell. We'll see examples of that in a second. The opposite of that is endocytosis, and this is a situation where a cell needs to take in large molecules and form a vesicle. There are three types of this: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is essentially cellular eating. You have cells in your immune system called macrophages which do this, and what they do is they go around and they pick up foreign bodies, take them into themselves via vesicles, and then they destroy them. Here's what that looks like. So here's phagocytosis. And let's say this is an amoeba or a single-celled organism that's eating. It's going to surround the food with these little extensions called pseudopodium. And what eventually is going to happen is the membrane is going to fuse around that piece of food, and it's going to form a vacuole. And therefore, it's going to take that food particle into the cell without ever disrupting the cellular membrane.